Hello everyone, welcome back to the session. So in today's class we are going to discuss about protein. In the previous class we have discussed about amino acids, okay? So we'll continue the biochemistry and today topics will be the protein. So as I said before, yesterday's class we have completely dealt about the amino acids, and in today's class we are going to start a protein, and yesterday's class and today's class it's somewhat similar to each other because group of amino acids is nothing but a protein, okay? We'll discuss it. So how can we define a protein? Proteins are high molecular weight biomolecules composed of one or more polypeptide chains made up of amino acids linked by a peptide bonds. It is a higher molecular weight, and the group of amino acids is a low molecular weight, which is in low molecular weight. So many amino acids that make up a bulk compound, bulk molecule. So that is why we are saying protein as a high molecular weight biomolecules composed of one or more polypeptide chains. So in yesterday's class we have discussed about what is amino acids, what is peptide bond, and what is peptide, and what is polypeptide chains, and what is protein. So in yesterday's class we have discussed the difference between all these terms. If you not watch the class, just go through the class and come back to the session. So makeup of amino acids and that linked by a peptide bonds. So one amino acid is linked with the another amino acid with the help of the peptide bond. So the peptide bond will be formed formed as a result of a condensation reaction. So that is important. Now this protein is derived from the Greek word proteos, isn't it? So the protein is derived from the Greek word proteos, meaning primary or of first importance. Of course the primary is playing a major role in the human body. So this protein is essential for a structure, for a metabolism, for regulation, for defense. So for all these processes we definitely need a protein. Now what will be the basic structure of a protein? So as in yesterday's class we have discussed about how many amino acids are present. So we are having approximately 300 different types of amino acids, but you should know the most common type of amino acids were the 20 amino acids. So in that 20 amino acids we are having two different types of amino acids. One is essential amino acid and another one is non-essential amino acid. So in the essential amino acid we need, that will be what will be the difference between this essential and non-essential. Essential in the sense that we need in the diet. Non-essential means our body will synthesize that amino acid, so don't need to take that type of amino acid in the diet. So this is what the difference. And also you should know what will be the what are all the amino acids are coming under the essential amino acid, and what are all the amino acids are coming under the non-essential amino acids. Now all those amino acids are linked by a bond, and that bond is called as a peptide bond. Very, very important. And the sequence of amino acid determines a protein structure and function. There is a sequence of amino acids. So many amino acids, they are linked by a peptide bond, and many polypeptide chain. Together we are having a high molecular weight, which is nothing but a protein. Now how the peptide bond is going to form. So in the last class itself I have shown you this picture. So this is what one amino acid. So we have a central carbon atom, and we have a hydrogen, and we are having an R group, and we are having an amine group, and we are having a carboxyl group. And this amine group, this is nothing but an N terminus, and this is a C terminus, and this is an amino group of another this is the second amino acid. Now see here, the carboxylic group of one amino acid and the amine group of the another amino acid, they are making they are react with something. So here an OH, and here a H. So this H2O will be eliminated as a water, and as a result we are having a peptide bond. And the peptide bond is nothing but it looks like this. So this is what a peptide bond. So this is very important. So this bond makes up a protein. So this is how the peptide bond will be formed between two amino acids. See here, they are formed between a carboxylic group of one amino acid and the amino group of the another amino acid. The reaction the reaction of these two amino acid form is condensation reaction. Why we are calling this reaction as a condensation reaction? The water molecule is getting eliminated. So that is what a condensation reaction. Now, so what will be the characteristic features of the protein? So the important characteristic features will be the planar and the rigid. Planar in structure and rigid. Okay, so this is the structure of the protein, and partial double bond character. Partial double bond character. So we are having a like this. So this is what a partial double bond character, no free rotation. So this is very, very important. Protein have no free rotation. Now, how the proteins is getting classified? The proteins are classified into, according to their structure, we are classifying the protein into three different types. One is simple protein, and another one is conjugated protein, and the third type will be the derived protein. So just like in the lipid. Now, so this protein, the protein is formed. So those protein which are on hydrolysis, they are giving only amino acids. So in the carbohydrates, those sugars on hydrolysis, they are giving a monosaccharide. We are having those sugars which are not hydrolyzed, we are calling them as the monosaccharides. Those sugar on hydrolysis, if it gives two simple sugars, we are calling them as a disaccharide. So based on that hydrolysis process, we have classified them into different types. So like in the same way, on hydrolysis, if the protein gives only the amino acids, we are classified them under the simple protein. Now, this conjugated protein on hydrolysis, in this protein, as the name conjugated, they are having a protein part as well as the non-protein part. If they are having both protein as well as the non-protein part, we are calling them as the conjugated protein. Now the derived protein, they produced from other proteins. So if any proteins, from that protein, one protein will be produced. So that is what a derived protein. So based on the structure, we are classifying them, classify the protein into three types: simple protein, conjugated protein, and derived protein. Simple protein, the protein which on hydrolysis gives only amino acids. That is the simple protein. Conjugated protein, again if the protein on hydrolysis gives a protein part as well as the non-protein part. So this is what a conjugated protein. And the derived protein, from the protein will be produced from some protein will be produced from some other protein. So this is what derived proteins. Now the same thing, now you should know what are all the examples, which proteins are coming under which classification of protein. So first type will be the simple protein. So as we mentioned before, on hydrolysis, they yields only the amino acids. So this is what the simple protein. And what are all the examples? Albumin, globulin, histones, protamines. So these are all the examples of the simple protein. So albumin on hydrolysis, they give only amino acids. Globulin on hydrolysis, again they'll give only the amino acids. Histones, again on hydrolysis gives only the amino acids. And protamines, again on hydrolysis, gives only the amino acid. So this will be all these proteins will be coming under the simple proteins. Now, so this is what the primary structure of a protein. So in the protein, we are having a primary structure, secondary structure, and protein tertiary structure. And all these simple protein are in a primary structure. We can say like that. Okay, so just know, or not all the simple protein. So basically we are having three different types of structure of protein, that is primary, secondary, and tertiary, and quaternary. So in yesterday's class itself we have discussed all these things. Now in the primary structure, they are having a group of amino acids. So the group of amino acids join together with the peptide bond. So this is what a primary structure. So this is linear in structure. Okay, all right. So the second important type will be the conjugated protein. So in the conjugated proteins, these proteins contains a protein part as well as the non-protein part. So these are all the conjugated proteins. So this non-protein part of the protein, non-protein component is called as a prosthetic group. Very, very important. Kindly make a note of it. So in the conjugated protein, we have said that the protein part as well as the non-protein part. So together we are calling them as a conjugated protein. Now this non-protein part, that will be, it is just called as a prosthetic group. Now we are having glycoproteins, which means what? Here protein is the protein, as we know, that group of amino acid. And what about this glyco group? Carbohydrates. So this carbohydrates will be associated with the protein. So that's why we are calling them as a glycoprotein. So this is what? A prosthetic group. So we have said that this is the protein part and this is the non-protein component. So the non-protein component is called as a prosthetic group, and here the prosthetic group will be the carbohydrate. And what this type of glycoproteins will be coming under which category? They are the immunoglobulins. Immunoglobulins are nothing but an antibodies. And lipoproteins, here the protein part is, there is a protein part and we are having a non-protein component, that is lipid. So lipid is associated with the protein, and the example will be the LDL and the HDL. High density lipoprotein, low density lipoprotein. And a chromoprotein. So we are having a protein part and we are having a chromo part. So chromo is nothing but they are the one giving the pigment. So here the prosthetic group will be the pigment. And what will be the example? Hemoglobin. Hemoglobin gives a red color. Heme part and the globin part. Okay, so this is very important. Now the metalloproteins, here the prosthetic group will be the metal. So the metal is a non-protein component, so that's why the prosthetic group will be the metal ion. And so these metalloproteins will be nothing but a ferritin. So inside the hemoglobin we are having a central metal atom, heme, that is ion, Fe, it is present there. So phosphoprotein, so phosphate group here will be the prosthetic group in the casein protein. So casein protein, we are having a protein part as well as the phosphate group. So that is why the example will be the casein. Now based on the shape of a protein, we are having a fibrous protein and globular protein. Based on the shape of a protein, we have two different types of shape of protein. One is fibrous protein and another one is globular protein. So how the fibrous protein will be? It is long and it is thread-like and it has a structural role. The fibrous protein have a structural role, and the example for the fibrous protein will be collagen, keratin and the elastin. So all these were considered as a fibrous protein. See, look at the picture. This is long and it is thread-like. So the thread-like protein is the fibrous protein and the globular protein. So this is what a globular protein. It is spherical in nature. It is spherical, it is compact and it has a functional role. See, the fibrous protein, they are having a structural role, and the globular protein, they are having a functional role. Functional role. So kindly make note of that, okay? So the examples of this globular protein will be the enzymes, hemoglobin and antibodies. So all these are having a functional role because antibodies have to fight against the antigen. There is no way it is contributing to the structure. So the globular proteins, enzymes, hemoglobins and antibodies were coming under the globular proteins. Now, so levels of protein structure. Just now we have studied the primary structure. It is the linear structure of amino acids. So the linear structure of amino acids will be considered as a protein structure, and these determined by the genetic code. So we are having 64 codons, and in the 64 codons, 61 will be the sense codon and remaining 3 will be the nonsense codon. So these will be the genetic code. So, and you should know that if there is any mutation in, so usually this is consider this as an mRNA. And this is a 5' end and this is the 3' end. And this is an mRNA. The mRNA has been synthesized just now, and this is the first codon. 3 bases will be there in the first codon, and the 3 bases will be in the second codon and the third codon, fourth codon, fifth codon, and then the sixth codon. So in the sixth codon, usually the bases will be GAG. Okay, so if the GAG in the A in place of A, the A has been mutated to T. GTG. So this GTG, usually this codon GAG codes for the glutamic acid. Glutamic acid. And this glutamic acid and this mRNA is meant for to maintain the structure of a hemoglobin. Structure of RBC. So this is, so instead of this glutamic acid, the A has been mutated to GTG. So this GTG codes for a valine. So that's why if any amino acid, if any point mutation has been taking place in one, even in one amino acid, the structure of, originally the structure of amino acids will be like this, but it will be changed into a sickle cell shape. So that is why we are saying a sickle cell anemia. So glutamic acid is replaced by valine in the hemoglobin because of the change in the one particular amino acid. Right? So this is what a protein structure, and if there are any mutations in the primary protein structure, again it leads to certain disorders. Now we are having a secondary structure. So in the secondary structure, the folding due to hydrogen bond. So in the previous class itself, I have mentioned you that, so there will be no specific bond, they are stabilizing the primary structure, but in the secondary structure, hydrogen bonds are involving in folding the secondary structure. So we are having two different types structure which is involved in the secondary structure, that is alpha helix and beta pleated sheet. So I have shown you the picture in the last class itself, and the alpha will be like this, alpha helical structure, it will be like this, and in the alpha helix those structure has been stabilized by the hydrogen bonds, and in the beta pleated structure it is like a sheet, it is like a sheet like structure, and it again it will be stabilized by interchain hydrogen bonds. Here it will be interchain and here it will be intrachain. So this is what the difference between alpha helix and beta pleated sheet. So in the secondary structure, these two structures have been involved. Now in the tertiary structure, so in the tertiary structure, so in the yesterday's class itself I have explained you about the tertiary structure, the overall 3D conformation of a single polypeptide. So this will be the structure of a tertiary protein which is in the tertiary structure. They are giving a 3D dimensional view, and the bonds which are involving in stabilizing the structure, tertiary structure, is disulfide bonds, hydrogen bonds, ionic bonds, and hydrophobic interactions. So all these bonds are important to stabilize a tertiary structure. What are all the bonds? Disulfide bonds, hydrogen bonds, ionic bonds, and hydrophobic interactions. So all these bonds are stabilizing that. Now quaternary structure. So this quaternary structure association of two or more polypeptide chains, polypeptide chains. Say for an example, hemoglobin, no doubt in that we are having a 4 subunits in the hemoglobin. In each subunit are a different polypeptide chain. So that's why we are saying a group of polypeptide chain, we are calling them as a quaternary structure. And in the best example will be the hemoglobin, and that has a 4 subunits. So the question can be asked that hemoglobin have 4 polypeptide chains. This can be expected in the exam. Now what are all the properties of a protein? The first and most important properties of a protein will be amphoteric nature. So the protein which are amphoteric in nature, in the sense they are saying that they can act as both acid as well as base. Why? Because of the presence of a carboxylic group as well as the amino group. So as both group are present in the same structure. So in a previous class itself I have explained you, carboxylic acid is donating the proton, and the amino, the NH2 group is an accepting a proton. Okay, so that is why if it is donating a proton, that can be divided, that can be act as an acid. If it is an accepting a proton, that is acting as a base. So this amino acid can act as both acid as well as base. So that's why we are saying this protein can act as amphoteric in nature. due to the presence of amino and carboxylic group. Very, very important. Now isoelectric point. So in the isoelectric point, so in the previous class itself, I have explained you the alanine and the protonation and deprotonation concept. So at a particular pH, no protonation will be taking place, no deprotonation will be taking place, which may it's not no, both protonation as well as deprotonation will be taking place, as one is acquiring a positive charge, another is acquiring a negative charge. So plus 1 minus 1 is equal to 0. As a result, there will be no net charge. So that's for that particular point where the net charge is 0, that point we are calling them as the isoelectric point. If you are having any doubts with respect to this isoelectric point, I have explained in the previous class itself very clearly. Kindly go through that. Now, solubility of a protein. So the solubility of a protein depends on certain factors, that will be pH, temperature and ionic strength. So definitely protein requires a proper pH. Okay, say for an example, an enzymes is nothing but a protein. Some enzymes requires an optimum, not some enzyme, most of an enzyme require an optimum temperature and optimum pH, only then it will not undergo any denaturation or renaturation. Okay, so if an enzyme or if any pH want to function properly, it definitely depends on factors like pH and temperature as well as the ionic strength. So all these are very important factor the solubility of a protein will depends on. Now, so the most important thing will be the denaturation of a protein. So what is mean by denaturation? So here initially consider this as a protein. Initially it will be like in this structure. So the laws of native structure and biological activity without breaking the peptide bonds. Now this structure, this box like structure has been changed into rectangle or triangle, whatever it is. Just imagine, I'm just saying for an example. So the native structure has been changed. The native structure has been changed and it will be arranged like this. Just see here, originally the protein will be like this, in this shape, and now without changing any bond, without breaking any peptide bond, just the structure has been changed like this. No protein will be in a triangle structure and no protein in a rectangle or in a square shape. Just for an example, I am saying this will be the native structure of a protein. Consider this, and this have certain biological activity. And now their structure has been lost as triangle, and and even though it has been changing the structure, there will be no peptide bond is breaking. As a result, they are losing their native structure as well as they are losing the biological activity. So without breaking any peptide bond, their activity has been disappeared. So that is called as a denaturation. This is important. So what is the difference between denaturation and renaturation? We often tend to confuse between these two words, that is denaturation and renaturation. Just look at this picture. This is a normal protein. This is a normal protein. Whenever the normal protein is changing their structure without breaking the peptide bond, and that particular structure is the denaturation. And if the denatured protein regained its original protein structure, and that process is called as a renaturation. Denaturation means it is losing their structure and biological activity and without breaking the peptide bond. So that is what a denaturation process. If a denatured protein regain its biological activity and the native structure, so that process is called as a renaturation. Very, very important. Just look at this picture. So this is a normal protein. So the protein has been denatured, which means changing their structure, but no bond is getting broke down. So this process is called as a denaturation. Now this protein which are denatured again gains its native structure as well as the biological activity, and that process is called as a renaturation. And one of the most famous example will be the egg. So whenever it is inside the shell, it will be in one structure. The protein which is in a native structure, after it has been uh, changing the structure, denatured. So this is what a coagulation of a white egg. If we are making an omelette, immediately after that the structure will be, the colorless fluid will be changed into white color. So that is what a coagulation of white structure. Very, very important. Now, so just imagine the egg example, and what are all the factors which are required, which have been experienced by a protein which are in a normal structure, it is undergoing a denaturation. What are all the factors are affecting to change the structure? That is heat. Again, we are breaking the eggshell and we are opening in it and we are pouring on the pan. So as a result of heat, it is changes to the white color. So the best thing will be the heat. And again, no doubt that will be the pH and the heavy metals, organic solvents and radiation. So all these were the causes of the denaturation process. Now, as a result of denaturation, what are all the effects which is not there in the original protein? So it is losing its solubility, losing their function and coagulation. So coagulation, the best example for coagulation will be the boiling of egg white. Now, what are all the functions of a protein? So the functions of protein, there is a structural function, collagen and keratin, so which is very important for the structure of a bone as well as a hair. So they are the structural proteins. And enzymatic proteins, so you should know all these proteins, all these structural, enzymatic, transport, all these were the proteins, but each proteins have their own function. So and the first function will be the structural protein, and the collagen and keratin will be the example. Now the enzymatic proteins, so enzymatic proteins, we are having an amylase and pepsin. Amylase and pepsin, so this is considered as the enzymatic protein. Now transport protein, so in the transport protein, the example will be the hemoglobin. And the hormonal protein, and which means hormones, again it is a protein. Those proteins which are functioning as a hormones, the example will be an insulin. Immune protein, so antibodies, that is a globulin protein, immunoglobulins. And storage, ferritin is responsible for storage. And movement, actin and myosin. Regulatory proteins, all the transcription factors, all those were coming under the protein. So these are all the functions of protein, very, very important function. Now, so protein digestion. So if protein, we are intaking a protein, or the certain amino acids are synthesized in our body itself, how all these proteins are getting digested, this can be expected in the exam. So how all these proteins are getting digested? Because of the enzyme. So if there is an enzyme which is responsible for breaking and digesting a nucleic acid, we are saying that as a nucleases. If the enzyme which are digesting the lipids, we are calling them as a lipases. If the enzyme which are breaking the protein, we are calling them as a proteases. Again, all these digestion will be taking place with the help of an enzymes. So so in the stomach, we are having a protein, that is pepsin. So pepsin converts protein into peptides. So just very important, a group of peptide, we are saying that as a polypeptides, and for many polypeptides we are saying that as a protein. So initially the protein will be breaking down into peptides. Now in the small intestine, these peptides is getting converting into an amino acids with the help of a trypsin, chymotrypsin and carboxypeptidases. All these enzymes are responsible for converting these peptides into amino acids. Now, so this amino acids will be the simpler form of a protein. We know that further these amino acids can't be break down. Now these amino acids are getting absorbed into the bloodstream. Right? Now, what are all the important structural motifs of a protein? So helix, turn, helix. So this like this. And zinc finger, leucine zipper, beta barrel. So all these were considered as a structural motifs of a protein. Now, what are the important examples and what will be their function? Hemoglobin, and what is the function of a hemoglobin? Oxygen carrying molecule. And insulin. What about insulin? Again, it is a hormone. Collagen. Collagen is responsible for the connective tissue. And keratin, hair and nails. And actin and myosin, muscle contraction. Very, all these are the important proteins. These proteins plays a crucial role in our body. Now, so a peptide bond is an amide bond, no doubt in that. Peptide bond is also called as an amide bond because of the involvement of amino group. Now, hemoglobin has a quaternary structure. Of course, if the protein has one or more polypeptide chains, we are saying that as a quaternary structure, that is a hemoglobin. Collagen, we are having two different types of protein based on the shape, and fibrous and globular, and this collagen is a fibrous protein. And enzymes are globular proteins. This also can be expected in our exam. And denaturation affects secondary, tertiary, quaternary structures, and primary structure remains intact in denaturation process. Primary structure remains intact in denaturation. Now, so that's all we have completed in the yesterday's class also. In the previous class also, we have discussed about the amino acids and proteins. So we'll discuss some of the questions. So which of the following bonds is primarily responsible for stabilizing the secondary structure of a protein? No doubt about that, that is hydrogen bond. Now, the peptide bond has partial double bond character due to the resonance between the carbonyl and the amide and nitrogen. So as a result of this we are having a peptide bond. So the same thing. And which amino acid contains a thiol group? So you should know that thiol, in the sense there is an involvement of ESH. Yes, sir. And sulfur containing amino acids were cysteine and methionine. Now, among these two amino acids, one amino acids are involving in the disulfide linkage. So that is nothing but a cysteine. So as a result of the bond and reducing agent, cysteine undergoes a reduction reaction. As a result, they are gaining a hydrogen. So they are forming a SH group, that is a thiol group. Now, at the isoelectric point, that is Pi, a protein has a maximum solubility, has no net charge, is completely denatured, becomes negatively charged. So at the isoelectric point, in the sense, one is deprotonated and one is protonated, and here carboxylic acid is donating a hydrogen and they are acquiring a negative charge, and the amino group, that is NH2, it is accepting a hydrogen which is released by the carboxylic group, so that it is gaining a positive charge. So as a result of this protonation and deprotonation, the net charge will be -1 + 1 is equal to 0. There have been no net charge. So as a result of that, we are saying this particular point as an isoelectric point where we have no net charge. So the answer for this question will be no net charge. And the fifth question will be, which level of protein structure is directly determined by the genetic code? So based on the genetic code, the level of protein has been determined. So that is nothing but a primary structure, no doubt about that. It is linear in nature. Now, which of the following proteins have quaternary structure? So quaternary structure is nothing but a group of polypeptide chains. Together we are calling them as a quaternary structure, and the best example for quaternary structure, and that is nothing but a hemoglobin. Hemoglobin is a protein which is having a quaternary structure, and originally this hemoglobin having a 4 polypeptide chains. Now, denaturation of a protein, it results in the cleavage of a peptide bonds, loss of a primary structure, loss of secondary and tertiary structure, and formation of new peptide bonds. See here, in denaturation, there will be a loss of a structure. No doubt about that. The native structure has been lost. So the native structure has been lost, and the function has been lost. The biological activity done by this native protein has been lost, but the peptide bond will not break. So that's why the denaturation of a protein results in a cleavage of a peptide bonds, no It is wrong. And the loss of primary structure, loss of secondary and tertiary structure, and formation of new peptide bond. So the answer will be loss of secondary and tertiary structure. So all the structure has been lost. Now, which of the following is a basic amino acid? So, in yesterday's class itself, I have explained you what are all the amino acids which are basic and what are all the amino acids which are acid. So, as what are all the amino acids are hydroxylic amino acids. So, as the name suggests, this aspartic acid and glutamic acid ends with an acid. So, it is an acid amino acid. Now, serine, threonine, all these were the hydroxylic amino acid or aliphatic amino acid. So, the answer will be lysine. Arginine, lysine, histidine. So, all these were the basic amino acids. The experiment that demonstrated the primary structure determines the tertiary structure of a protein was performed by. The experiment that demonstrated that the protein structure determines the tertiary structure of a protein was performed by. So here the protein structure has been converting into the primary structure has been converting into the tertiary structure, and that experiment has been done by which of the following scientists? So that will be the Anfinson. Now, which of the following interactions is strongest in stabilizing the tertiary structure? So, the tertiary structure has been stabilized by which bonds? See, the hydrogen bonds, hydrophobic, disulfide, van der Waals force. All these forces are involving in the tertiary structure. When compared to all these things, which is considered as the strongest? No doubt about that. That is the disulfide bond. So, as far as of now, we have completed the cell biology and we have started the biochemistry. In the biochemistry, we have completed the carbohydrates, lipids, and the proteins and amino acids. So the major biomolecules has been discussed. So we'll meet you in the next class. Thank you so much.